Hi everybody, welcome back to Neuronotes. I'm Elisa from the Mission Brain podcast team, and today we will briefly focus on two emerging and closely interconnected mechanisms in traumatic brain injury: endothelial glycocalyx disruption and glymphatic system impairment, both increasingly recognized as early drivers of secondary brain injury.
Following traumatic brain injury, attention has traditionally been centered on neuronal damage, axonal injury, and intracranial pressure dynamics. However, recent studies from the last months highlight that microvascular dysfunction occurs very early, preceding overt blood-brain barrier breakdown and significantly influencing edema formation and long-term outcomes. The endothelial glycocalyx is a carbohydrate-rich layer covering the luminal surface of endothelial cells, composed of proteoglycans, glycosaminoglycans, and plasma-derived proteins. In physiological conditions, it regulates vascular permeability, shear stress sensing, and leukocyte adhesion, and anticoagulant balance. In the cerebral microcirculation, it represents a critical functional component of the blood-brain barrier.
Recent experimental and clinical evidence shows that mechanical trauma, oxidative stress, and inflammatory mediators rapidly induce glycocalyx shedding after TBI. This degradation increases endothelial permeability, facilitates plasma protein extravasation, and promotes vasogenic cerebral edema. Importantly, biomarkers of glycocalyx damage, such as SYNDECAN1, have been associated with injury severity and favorable neurological outcomes, suggesting a potential role in early risk stratification. Parallel to vascular dysfunction, growing attention has been directed toward the glymphatic system, the perivascular clearance pathway responsible for cerebrospinal fluid and interstitial fluid exchange and removal of metabolic waste. Glymphatic function relies on intact perivascular spaces and polarized astrocytic endfit, also aquaporin for channel distribution.
Recent data indicate that traumatic brain injury leads to acute and persistent impairment of lymphatic flow due to astrocytic disorganization, aquaporin 4 mislocation, and vascular structural changes. This dysfunction results in the diffuse clearance of neurotoxic proteins, including tau and amyloid beta, providing a mechanistic link between TBI and post-traumatic neurodegeneration. What has become increasingly clear is the functional connection between endothelial injury and glymphatic failure. Glycocalyx degradation alters vascular pulsality and mechanotransduction, which are essential driving forces for glymphatic circulation. As a consequence, microvascular damage directly transfers into impaired brain waste clearance.
From a clinical perspective, these findings emphasize the importance of early endothelial protection and optimized cerebral perfusion, alongside traditional neurocritical care targets. Strategies aimed at limiting inflammation, oxidative stress, and secondary systemic insults may indirectly preserve both glycocalyx integrity and glymphatic function.
In conclusion, recent studies reframe traumatic brain injury as a disorder involving vascular biology and fluid clearance systems, not solely neuronal and axonal damage. The endothelial glycocalyx and the glymphatic pathway represent key therapeutic and prognostic frontiers in modern neurotrauma research. This was Neuronotes, a technical update for Mission Brain. Thank you for listening.